Okay, so today I'm going to take a short look at section 5 in the syllabus, catalysts, and enzymes is also chapter 5 in the textbook. And what we're going to talk about here are enzymes and their role as catalysts in a biological system. A catalyst is something that speeds up a reaction, a chemical reaction, without being consumed itself. So for example, if you throw a Mentos into a Coke, we all tried that, that speeds up the release of the CO2 bubbles a lot. So in this case here, our Mentos is a catalyst. Now, enzymes are catalysts in the cell, and they're made of proteins. And if you remember from chapter 4, section 4, that all proteins are built of different amino acids. Now these amino acids can then fold into a secondary, tertiary, and even quaternary structure. And when that happens, these enzymes can get a three-dimensional shape that will maybe end up having some kind of fold in it. And that fold, when we're talking about enzymes, is what we call the active site. And the purpose of having an active site like this is it's going to fit perfectly to the molecule that the enzyme wants to interact with. So let's look a little more about the enzyme and the substrate. Again, this is how it's drawn in the book, but realize this is not how it looks in real life. In real life, these are complex three-dimensional structure with active sites of specific molecules. So just be aware that these are just a drawing. This is not reality. So here we have our enzyme. And here we have our substrate. So this could be inside the cell, where this substrate is something that is damaging to the cell, something we need to get rid of, so the enzyme needs to be able to break this down. And as we can see, there is a similarity here between the substrate and my active site. This is what we call the lock and key hypothesis, that the substrate fits into the enzyme like a lock and key. Again, reality is a little more complex than that, but it's a good idea for you to know. So our enzyme here and the substrate bind. So my substrate now, part of it fits into the active site of the enzyme. So now they have formed a enzyme substrate bond. Now my enzyme now helps to catalyze a reaction where the enzyme then breaks down the substrate, and the substrate is then broken down into new molecules we call products. So now we go from having the substrate to having the products. And since our enzyme is a catalyst, it is now ready to do so again. It is ready to break down another substrate into its products. Okay, let's look a little bit more at the different properties of the enzymes. All enzymes are made out of protein, amino acids, which are then constructed into secondary and tertiary, maybe in quaternary structures. These enzymes will work better as temperature increases. So if we have here a graph where we here have temperature, and here we have activity. Maybe we start at 0 degrees and go up to 100 degrees. So what happens is, as we have a low temperature, our enzymes are not very reactive. Because, again, low temperature means low movement of the molecules, so it's less likely that the substrate will fit into the active site because they're moving around less. So at a low temperature, not a lot is happening. As we increase the temperature, the enzymes will start working better and better and better because now everything is moving more, it's vibrating more, and all chemical reactions goes faster at higher temperature. But this will not keep on increasing because, as we said, all enzymes are made from protein. So protein will, at a certain temperature, the bonds will start to break down. It will denaturate. So there'll be a temperature somewhere where after that our enzyme will stop working because this is the temperature here where this protein denaturates. And in most humans, we know our enzymes tend to work really well around body temperature, but if the temperature gets a lot higher, it will break down. We can see this in meat, for example. Some meat, when it's uncooked, is quite stringy, but if you cook it for a while, it becomes really mushy. That is because the heat breaks down the proteins, break down the bonds. So the same would happen to our enzymes. So that is why if we boil something, the enzymes will stop working again normally. Because there are some enzymes that works at a much higher temperature than others. We have discovered animals who live at what we call hydrothermal vents, and they live in symbiosis with bacteria, and those bacteria can survive at a remarkable high temperature. So they will have a different curve. They will be able to tolerate a much higher temperature than normal animals. But keep increasing temperature enough and all enzymes will eventually denaturate and stop working. If we look at pH and activity of enzymes, this also has a huge influence. So most, if not all, enzymes have an optimal pH. This is a pH where they tend to function the best. So for example, an enzyme like pepsin, which works in our stomach, is very good at a low pH here too, because this is PUC, and then again its activity will decrease as the pH gets higher, while a enzyme like amylase, for example, might have a curve that looks something like this. So different enzymes have different optimum pH where they work the best. And when you think about it, it's quite smart that we can have one enzyme that works really good in the stomach, but when we get outside the stomach and the pH then gets higher, it stops working. When we talk about digestion, this will make a whole lot more sense. Just remember that here, if we then lower the pH again, the enzyme will start working again. So unlike temperature, where if the temperature gets too high, the enzyme is permanently damaged, if you vary the pH, you can vary the rate of conversion by the enzyme here, but unless you go like a really strong acid or really hot base, you don't destroy the enzyme, you just lower or stop the activity. So here an example from last year's exam, where they ask what is a correct definition of the term enzyme. And as they often do, they come up with a lot of words that almost sound correct, but there's only one right answer. So the first thing here is to say that enzymes are carbohydrates that works as biological catalysts. It's true that enzymes are biological catalysts, but they're not carbohydrates. Then they say, so it cannot be A. Then they say B, carbohydrates that act as substrates. Yes, carbohydrates can be substrates for the enzyme, but that doesn't make them the enzyme. So it can't be B either. Then it says in C, proteins, okay, now we're getting somewhere, that act as a biological catalyst. That is our definition of the enzyme, so it is C. But also in D, they say protein that act as substrate. Yes, proteins can act as a substrate for the enzyme, but that doesn't make them the enzyme. So our only correct answer here will be C. So here is another example from last year's multiple choice exam. And here it's a little more complex because we have to look at different things. We are told we have an enzyme here that digests protein in the stomach. And here we also need to remember that in the stomach the pH will be low due to hydrochloric acid. We're told that 4 test tubes were set up, each contained the same amount of protein and the enzyme X. We don't know the enzyme. The test tubes are kept at different level of pH and temperature as shown in the table. In which test tube will digestion be quickest? Well, first of all, we know this is in the stomach. So, because of that, it would be fair to assume that a pH of 2 will work better than a pH of 7 because enzymes in the stomach are adapted to the low pH. So, it must either be A or B. We can now totally forget C and D. So it's either A or B. Then we have to look at the temperature. There are 2 different temperatures here, 20 and 35. We do know that as we increase temperature, enzyme reactions speed up. In the human body, they will speed up to around 40 and 42 degrees. So A will go quite fast, but B, due to the higher temperature, will be faster. So the only correct answer here is B.